Alright, so this is part 2 of the lecture on enteroviruses and polioviruses. So, welcome back. If we look at the structure of poliovirus, remember it's small, it's naked, it's icosahedral. It has 4 structural proteins, but looking from the outside, you can only see 3. So VP1, VP2, and VP3 come together to form these triangles, and the triangles come together to form the pentameres, and then underneath this, so you've got all of these triangles coming together to make your icosahedron, the overall structure, but underneath, so VP1, 2, and 3 are on the outside, and that's just viral protein 1, viral protein 2, viral protein 3. Underneath, viral protein 4, VP4, forms kind of the underlying scaffolding for the capsid. And then inside, you've got your small genome. It's positive single-stranded RNA. Now, here's the electron micrograph of the viral particles, and then here's the model, the cartoon. These viruses are incredibly stable. These viruses are resistant. Just a moment. Okay, so as I was saying, it's very, very stable. Very stable in the environment. It's stable at low pHs. It's stable against most enzymatic attacks. It's stable against detergents, drying, other disinfectants, and this can last weeks on surfaces. So with a lot of enveloped viruses, it might be stable for a few hours. This naked virus here can be stable for weeks. So it's difficult to control the spread. In order to inactivate it, you need very long wet times or very long contact times. So you put the bleach on the surface and you let the bleach sit there for a long time. Heating up, you can heat it to 50 degrees Celsius for an hour, and that denatures the proteins, but you need to do it in the absence of these cations because they can help stabilize it. So this is a really tough little virus. So how does it cause infection? How does it make you sick? So poliovirus can only infect humans and non-human primates. There's no other reservoirs, like bats or anything like that we have to worry about. Now, this actually makes studying it a little bit difficult because you can't just infect mice with it easily, so it makes it a little bit harder to study. So this enters orally, and it replicates in the tonsils. And then from there, the next site, it replicates in the Peyer's patches. And so this is the the gut-associated lymphoid tissue. So in your small intestine, you've got so in this picture here, this would be the lumen right here, and then you've got the epithelium, and then underneath the epithelium with the microvilli and so forth, you've got these follicles. You've got these specialized lymph spots, these tissues with lots of immune cells. And so this virus likes to replicate in that immune tissue, so either in the tonsils or in the Peyer's patches. Now, for most people, it's going to end there, and then your immune system will eventually take it out. But for some people, it spreads into the blood, so that's viremia. And then it can spread to other areas. Now, you can get a sore throat, fever, headache. If it spreads to your spinal cord, that's when you can get the paralysis. Now, the virus, if we look at its replication cycle, its genome and its replication cycle. So first, Like all viruses, it has to attach, and attaches to a poliovirus receptor, and they actually call it this because they first discovered this protein, this surface protein, as the poliovirus receptor, so that's its name, poliovirus receptor. Now we know it as CD155, which is not super helpful, so CD is cell differentiation factor 155, so immunologists just name things, number them, but this protein is important. It has a normal everyday function. It's not just a doormat for poliovirus. It's important for stimulating natural killer cells. Other enteroviruses can have more than one receptor, and they can have a slightly broader tropism. So the virus enters through endocytosis. It uncoats, and its genome enters the cytoplasm. Now, if we take a look at its genome, we've got a structural region up here. We've got the internal ribosomal entry site, and then you've got two segments in the genome. We've got the structural region and the non-structural region. And when this gets translated, so it's a positive sense, so it can act as a messenger RNA. So when this gets translated, you get this big polyprotein. And it's very important that you have proteases that cleave it so you get your separate functional proteins. So the first protease actually kind of auto-cleaves itself out, and then it's able to cleave the rest of the polyprotein into the functional proteins. We'll talk a little bit more about the details of that. Your structural proteins. So structural proteins are always going to be your capsid. And the non-structural proteins are going to be proteins that are essential for replication that don't make up the capsid. So looking at the next slide here, if we zoom in on the 5' end, this little circle here, this is a small viral protein that's associated with the genome, so VPG. And this protein has to be associated with the 5' end of the genome in order for a proper replication. We'll talk more about that during the replication cycle when we go through it, and when it's useful, when it's essential in just a couple minutes. Now, the internal ribosomal entry site is really important. This was actually, I think, the first or one of the first irises that were discovered. And this iris allows cap-independent translation. And this is important, the structure of this iris, because some of the proteases in the during infection, one of the viral proteases, or viral protease 2A, is going to cleave eukaryotic initiation factor 4G. When this happens, the ribosome can no longer recognize capped cellular messenger RNAs. So all cellular translation essentially shuts down because you don't have a working EIF4G. So that means the only translation that can occur is through the iris, which is pretty cool. That's actually pretty clever. Now, looking at the genome, so this is the 5' end, and then we've got our, in here, the rest of the genome. And then the 3' end is polyadenylated. So we've got it comes with a poly-A tail. Now, during the replication cycle, so we're going to attach to the poliovirus receptor, come in through endocytosis, uncoat. We start translating the genome right away. That first protease auto-cleaves itself out, and then it cleaves the polyprotein into the functional protein subunits. Now, the virus is going to reorganize the membranous organelles inside the cell, and it's going to form a viral factories. Now, this is important because it needs to kind of hide itself within these membranous structures, within these kind of membranous vesicles, because it doesn't want to activate the innate immune system. So it's using these to protect itself, to protect the double-stranded RNA intermediate. Remember, it's a positive sense RNA. In order to replicate, it has to make the negative sense, and then it can run copies of the positive sense off of that. But during replication, you've got a temporary double-stranded RNA intermediate that could set off the innate immune system. So one of the reasons that it kind of reorganizes these membranes is to protect that double-stranded RNA intermediate from the innate immune system. So VPG, the viral protein that's associated with the genome, it's essential for the replication of the RNA genome. So the RNA polymerase needs a primer to start. So the VPG gets a whole bunch of U's attached to it. It gets polyuridylated. So you get a whole string of U's, and then that works as the starting point for the polymerase. So we make lots of copies of our genome. We make lots of copies of our structural proteins, VP1, 2, 3, 4. They're going to self-assemble, just like in the little shaking jar. All of those subunits self-assemble, and then the viruses escape. Now, the viruses can leave through exocytosis. And they might have, you might have like a cluster of viruses inside a membrane, but most of the viruses, you're going to get, they're released when the cell lyses. And so you're going to get a lot of naked viruses leaving during the death of the cell. After cell lysis. Okay, we'll end with treatments and prevention in the next segment. So this is part 2 of 3.